OK, so I guess I'll make this informal since it's just the 5 of us. But I'm going to talk today about mainly conductive hearing loss and Alagille syndrome. And this is something that has largely not been investigated very well before. And I should say, I got in this kind of in a very unusual way, rather than kind of setting out and saying I'm going to study hearing or I'm going to study Alagille syndrome. I'm actually a developmental biologist who studies how the head forms during development and where the genetics of why all the different bones in your ear have the correct shape so that everything can function properly. And so really, it's from studying animal models, first zebrafish and then mice, that we started to become more curious about whether, so these are models that have Jaglb mutations in both zebrafish and mice. And they have very particular defects in the bones of the middle ear. So we started to be interested in, well, how common could this be in Alagille syndrome? Because there's actually a few older reports suggesting middle ear problems in some Alagille syndrome patients. But there's almost no data out there on, good? OK. OK. There's almost no data out there on how prevalent it might be. So I'll just kind of take you through a story of how we got here, and then a few words at the end about how it might directly influence people with Alagille syndrome. So this is the mammalian, or more specifically the human, middle ear. And the way sound is transduced is it comes through the outer ear. Vibrations are collected in this auditory canal. And then it vibrates the tympanic membrane. And the role of, and within this space here, there's three of the smallest bones in the human body, called the malleus, incus, and stapes. And these form a very precise chain of bones. And their role is to transmit vibrations from this tympanic membrane to the cochlea. And so air comes in here, this tympanic membrane vibrates, which vibrates these little ear bones. And then what the ear bones does, particularly through the stapes, is it then vibrates this cochlea here. And it's within the cochlea that you have the specialized neurons called hair cells that detect the vibrations and then transmit this to your brain. And that's how you hear. So essentially, this looks like a spiral, kind of a snail shell. And all along this snail shell are different neurons that have a preference for different frequency of sound. So very low frequency sounds, really deep sounds are here, and then the very highest frequency sounds are down here. So really, these middle ear bones play a really important role in allowing sound to be properly amplified to get to these neurons that can then detect that sound. And so really, the hypothesis is that some forms of hearing loss in Alagille syndrome may be conductive in nature, involving these middle ear bones. And this is, as I'll tell you, motivated by work on what Jagged does in other vertebrate species. And so I just want to I don't know how many of you are familiar with zebrafish at all? You are? Zebrafish research, or just zebrafish from?
High school biology.
Yeah. So most of you probably know about mice and rats, which are used very often for looking at human disease. But really in the last 10 or 20 years, zebrafish has become a much more prominent model. And so this is not really a niche field. There's over 1,000 labs now worldwide studying zebrafish. And it's growing every year. So it's probably one of the fastest growing model organisms. And it's starting to rival mice in terms of the number of researchers working in it. And the reason people study these zebrafish, there's some practical reasons. So they breed quickly and often. So for example, one pair of these fish can lay over 200 embryos per week. So you can get a lot of embryos. And they're very, the adult fish is about that big. And so you can really grow large numbers of fish at a very low cost. So for example, in my lab, we have 3,000 tanks of fish, and each tank has about 15 to 20 fish in it. So we have close to, I guess, 50,000 fish. And my lab is not is a medium-sized lab for the community. And the reason it's interesting to people is it's not like a fruit fly or a worm that is also another study. It's a vertebrate like us. So almost all the same organs we have, fish have. They have bone and cartilage. They have hearts, liver, pancreas. Only, for example, they don't have a lung because they're in the water. So there are a few organs they don't have. But they're very similar to us in terms of the types of organs. And we're increasingly understanding that genes that are disrupted in mice or humans that cause organ defects, you get rid of that gene in zebrafish, it often affects the same organ. So the idea is that it's another system that we can use to really understand when you're losing a gene, why do you get the defects that you do in particular organs. There's other, for developmental biologists, people like to study these because unlike mice, the embryos are laid directly in the water from the one-cell state. And zebrafish in particular are completely see-through. They're completely transparent up to a couple days of development. So it's really easy to directly observe all the cells in the embryo and how the cells come together to make different organs, and then what goes wrong in the assembly of those cells when certain genes are lost. It also develops very quickly. They hatch out of an eggshell in 2 days, but what's more important, from fertilization, just 4 days later, they're already swimming around and feeding. So things happen really quickly, so you don't have to wait for weeks and weeks for an organ to form. Also, its genome has to be completely sequenced, and it has most of the same genes that we have. And it's very easy now to delete specific genes. So just take you through a few concepts of how fish are actually more similar to us than a lot of people think. And this was work about 150 to 200 years ago by this German guy, von Baer. But it really kind of drove home the point that if you take, even though like a human or a chicken or a fish look really different from one another, if you look at embryos in development of each of these vertebrates at a particular stage quite early and pull these out, these look remarkably similar. So it's as if all these animals go through a common stage and then after that begin to diverge. And it's at this stage when a lot of the primitive organs have already started to form. So you can imagine that if you want to look at real basic biology of how different organs like heart or liver or skeleton form, it's going to be very similar between the fish and human. This is just showing you what zebrafish development looks like. This is starting at the one-cell stage. So you can see this is all the yolk, so this is not the animal, but these are the cells that just synchronously divide. And then after about 4 hours, these cells just start to spread out over the surface of the yolk. And now by about 12 hours, you can already start to see the eye forming up here. Here's the eye forming, the different vertebrae, or what's going to be the vertebrae forming down here, and the tail. And this movie was made over 20 years ago. It's a fairly low resolution type movie. It just gives you a sense of how you can really visualize how development is happening and then understand how it might go wrong in certain situations. The other thing is you can find a lot of mutations in zebrafish, either spontaneous mutations or a lot of labs will treat these fish with a mutagen, meaning that you randomly mutate the DNA and then you can randomly mutate genes and you just pull out animals that look different, or embryos rather, that look different from others. I think there's now thousands of zebrafish mutants out there, but I find it quite interesting that the first ever lab mutant for zebrafish was called golden because these animals didn't have the nice black pigmented stripes that this type of wild type strain has. And it was kind of more reddish and golden in pattern. And so this was in 1981, so 30 years ago. And just a few years ago, they finally figured out what the gene was that was mutated in here. And when they looked at human populations, it turns out that differences in this gene, which you don't have to doesn't really matter what it is, accounts for 30% of human skin color differences. So I think it's just kind of a nice story of how zebrafish can be relevant for human biology as well. And then the zebrafish genome was very extensively sequenced and annotated recently. And so and these are conservative estimates, but 71% of all human genes have a clear homolog, almost a nearly identical copy in zebrafish. And if you look at human disease genes, this number goes up. So 82% of all human disease genes have a clear similar gene in zebrafish. So the first work trying to model Alagille syndrome in zebrafish was actually done by Michael Pack. He's at Children's Hospital of Philadelphia, which I'm sure you're all familiar with from the work of Nancy Spinner and others who come to this meeting. And at the time, this was before we could really make genetic mutants very easily. So he used a trick where he was able to essentially use a small piece of molecule that looks like a DNA that inhibits certain genes. And by doing this, he was able to get rid of several of these jagged genes or notch genes. And so this is a bit, sorry, I didn't correct this from his paper, but we now know that this, what's called 3 here, is actually jagged 1. And this J2 is jagged 2. And what you can see is that here we're looking at the bile ducts in zebrafish. If you get rid of both jagged 1 and jagged 2, you get a paucity of these bile ducts. And this is very similar to what's seen in Alagille syndrome liver, the intrahepatic biliary duct phenotype. And there's also a similar phenotype of the ducts in the pancreas. And it's also known that some patients with Alagille syndrome have pancreatic duct problems as well. And so I'm going to tell you now how we got into this field of jagged 1. And so as I introduced, my lab is very interested in how the face is patterned in addition to how cartilage and bone develop. And we look at some particular structures that form the jaw of zebrafish. And so this would, for example, be if this is a 5-year-old fish, everything in blue is stained for cartilage, and red here is bone. And at this stage, the fish is mainly cartilage. Later on, a lot of the cartilage will turn into bone. And what happens is that there's these very interesting cells called neural crest cells that form near the developing brain. But during early development, these cells move from the brain down to where the face is going to form. And then these cells then become the cartilage and bone that forms most of our head and most of the zebrafish head. And so this is just showing nicely how we can visualize these cells right after they migrated down, then turning into the jaw skeleton. So this would be the eye, this would be the ear, and then you can see these cells start to coalesce and form the lower jaw, the upper jaw. And then also what you can see is that these cells here, this is a skeleton here that then connects the jaw back up to the ear. And so we use this as a simplified system to understand, well, how does each of these cartilage and bones get their distinctive shape? And so the way we went about that is we just said, well, what if we randomly mutate genes? Which genes, when we get rid of them, will affect the shapes of these different skeletal elements? And we found one mutation that was quite interesting because it affected really only 2 of these skeletal elements. So you can see that this element here and here, their shape became very different when we lost this gene. And we later found out that this is a complete loss of function of the zebrafish equivalent of the jagged 1 gene. And so really this was our entry into studying jagged 1 function and the connection to Alagille syndrome. And so really the idea here is that there's kind of these structures form more the upper jaw or upper face, whereas these structures form the lower jaw or lower face. And what's happening in this mutant is that these structures here start looking more like the structures that are found in the lower face here. So rather than being this kind of plate-like structure, it's more of a small rod like this element down here. So what we did an additional experiment where we took the human jagged 1 gene and we made too much of this in zebrafish. And when we did that, we actually got the opposite phenotype. So if we now put in too much, in this case we're using human jagged 1 in zebrafish. So even though it's a human protein, it's functioning in fish. And these fish, what we find is now this lower face starts to look more like the upper face. And so this was very interesting for us as developmental biologists, because this was saying that the jagged 1 is really important in kind of making the upper face different from the lower face. And just kind of as a tangent, which we don't have any, well, really hard data, but our feeling is that there's this characteristic facial appearance in Alagille syndrome. And over time, I've talking to some of the doctors who regularly see Alagille syndrome patients. What happens is the lower jaw kind of grows more than the upper face. So we think that might mean because this Jagged1 has more of a role in the upper face, the lower face is growing at a slower rate than the upper face is growing slower than the lower face would. That kind of extends the jaw out more and may be one explanation for the difference in facial appearance. The other thing is, I don't know, really, I don't want to get into too much molecular detail for this audience, but just say that, in some cases of Alagille syndrome, Notch 2 is lost, even though it's much rarer than Jagged1. And it turns out in zebrafish, actually, we have to get rid of both Notch 2 and Notch 3, and then we get the same phenotype as getting rid of Jagged1. So one idea, again, not really tested, is that the reason not a lot of people have Notch 2 mutations is that most often Notch 3 can kind of compensate for Notch 2. And I just say that we think part of the way that Jagged1 is making this upper face different than the lower face is that it's actually preventing skeleton from forming too early during development. And so really in this purple stain here, this is marking cells that are just beginning to form cartilage in the upper face. What you can see in the Jagged mutant is now there's more cells inappropriately making this BARX1 gene that's in pink here. And so we have a lot of work on the kind of the detailed molecular mechanism by which Jagged1 is patterning the skeleton, but I won't get too much into that. But just to kind of schematize that, we think that really what the role of Jagged1 in the face is kind of restricting the amount of cartilage that forms so that there's enough cells left later on to form the right amount of bone. And so it turns out when you get rid of Jagged1 in zebrafish is that this cartilage gets reduced. And then in addition, there's not enough bone cells here to make this large bone, and instead they make a smaller bone here. And so from here, I kind of want to transition more back to the middle ear, because I think that's probably what's most interesting here. And so really then, how do we go from the zebrafish jaw to thinking about the middle ear? What relevance might they have to one another? And this turns out to be one of what I think is one of the most fascinating stories in vertebrate evolution. So there's a really nice essay by, I don't know if anyone's heard of Stephen Jay Gould. He's a very famous evolutionary biologist who's written a lot of popular books. And there's this idea, ear-full jaw. And what does that mean? And essentially what it means is that during vertebrate evolution, what's happened is that what was once the zebrafish jaw, these bones have acquired a new function in mammals. So we have a completely different jaw articulation than fish do. And now what's happened is that the jaw bones of fish have now recessed up into the middle ear and are now functioning as those malleus incus stapes, those little middle ear bones that transmit vibrations that I showed in the first slide. And so there's a lot of embryological data suggesting that these are kind of the equivalent bones during evolution. And really just a schematic here, sorry if this, again, might be a little complicated, but you can see this bone right here and this bone right here. These are the upper and lower jaw of a fish. And they make a joint together, and that's the hinging jaw of a fish. But what's happened as fish have gone to reptiles, these bones have gotten a lot smaller. And now once you get to an early mammal here, what's happened is that this squamosal bone here and the dentary make a new jaw joint. And now what used to be these large fish jaw bones have now recessed up into the middle ear. And now because a new jaw has formed, they no longer need these old fish bones to be the jaw. And that allows them to now be repurposed to a new function. So I don't know if I want to get too much into this, because I'll skip that for now. And so really what we wanted to do now is saying, well, since we think that the middle ear bones are homologous to these jaw bones that are affected in a Jaglb1 fish mutant, is it true that if we get rid of Jaglb1 in mice, the middle ear bones will be defective, as we predict? And so the way we did this is, maybe I have to go back here to explain. So these are these neural crest cells that I talked about that very early during development, they form right where the brains should form. But then what happens is they migrate way down here. I'm showing these cells moving down to the ventral part of the face. And then it's from these cells that most of your facial skeleton comes from. So what we can do in mice is just get rid of the Jaglb1 gene just in these migrating cells. And so just in these cells that are migrating down here, they're going to form the head skeleton. We genetically delete the Jaglb1 gene just in these cells. And so that's what we're doing here. And interestingly, these mice have a very characteristic facial appearance with a severe underbite and a broad forehead that you can see here, which is actually quite similar to what's been described in Alagille syndrome. And I should just point out that there's actually, there was another group while we were doing this work also reported the same sort of phenotype independently from Stephen Goody's lab at Vanderbilt. But we were, again, most interested in the middle ear bones based on this homology. And so what we did then is we took these mice and we dissected, we euthanized them. And then we stained their bone with a red stain, and then very carefully dissected out the little middle ear bones from their ears. And what you can see is that these two bones here, the incus and stapes, these are thought to be the evolutionary equivalent of the two jaw bones in fish that we found were defective when we got rid of Jaglb1 in fish. So we predicted that these were the two bones that would be affected. And indeed, we see defects in these two bones, in particular the stapes bone. So you see that usually the stapes is this, stapes I think is Latin for stirrup, or Greek for stirrup. And because it kind of looks like a stirrup if you ride a horse, it's got this kind of circular morphology with a footplate here. But in these mice that lack Jaglb1, and again, this is just in those neural crest cells that make the head skeleton, you can see that the stapes has this very filled in, narrower morphology here. Interestingly, we remember that Notch 2 also, and in rare cases, can lead to Alagille syndrome. So we also separately remove Notch 2 from these same neural crest cells. We get the same exact defect, where the stapes is very thin and filled in here. And then we actually looked at something that's probably a more accurate Alagille syndrome model. So we looked at mice that lack only one copy of Jaglb1, just like in humans. And interestingly, we see the same stapes defect that we see when we get rid of both copies of Jaglb1 or Notch 2 in these neural crest cells. However, in these mice, we only see this defect about half the time. So half the mice are completely normal, and then half the mice, the heterozygote mice here, have this filled-in stapes. We do see some very minor incus defects here, which don't show up very well in these pictures and are quite minor. So it's really the stapes defect that we found to be more prominent. So the next thing we want to say, well, does it really matter that this stapes is malformed? Maybe even though this is a bit different shape, sound can still be conducted quite well and it would not affect hearing at all. So what we did is we did hearing tests on these mice that lacked Jaglb1 in the neural crest cell that had this stapes defect. The way hearing tests are done in mice is a little different because you can't really ask them to do, it's hard to ask them to do tasks if they hear a sound in their left or right ear. So, and I should say we actually did this with a collaborator who works in a hearing institute at University of Southern California. And what they do is they play sounds, and with electrodes they're recording in their brain for characteristic spikes of electricity that are produced when these mice hear sounds. And essentially what this graph is showing is how much sound it takes to elicit electrical response in the auditory brainstem. And so the idea is that if you need more sound, like higher decibel sound, to get any sort of response, that means that these mice can't hear very well. So it's like having to shout at someone to get them to understand. And what we find is quite a pronounced hearing loss or hearing reduction in these mice. And so that's really consistent with this Stapes middle ear bone defect causing some sort of hearing loss in these mice. I should just point out that it's also known that Jagged1, so I've showed you that Jagged1 plays a role in these middle ear bones. And I should say we haven't, this is all unpublished data that we're publishing soon. But there's also some other mouse mutants out there that are spontaneous mutants. And one of them is called Alagille. And it has this vestibular defect, so they have a problem with balance. And it turns out that loss of Jagged1 also affects these semicircular canals that are involved in the vestibular system in terms of balance. Actually, several Alagille syndrome patients have been reported to have defects in these semicircular canals. In addition, in the cochlea, so this is where those neurons are that actually detect the different frequency of sound. It's known that Jagged1 also has a role in patterning the different neurons and supporting cells within this region here. And so really, in addition to what I'm telling you about the middle ear bones, there's also several other roles of Jagged1 in the ear, which could explain other forms of hearing loss. And so based on our first fish and then mouse work, we really thought that there could be Alagille syndrome patients out there who had defects in their middle ear bones that could be causing some sort of level of hearing loss, though we didn't really understand how much hearing loss that would be. So we were able to dig up a few older papers. So there's very little out there in the literature, but we did find one paper in 1990 that was in a study in Japan where they I think they had looked at 2 patients post-mortem and dissected out their temporal bone with the middle ear bones. And in these 2 patients, one of the patients was said to have a bulky stapes, and then the second patient had both a bulky incus and a bulky stapes. They didn't publish any of their pictures, so I don't really know what this looks like, but at least there's then precedence for this. In addition, there was, again, a different study from France around the same time where they looked at one patient who had a form of hearing loss that looked both conductive and sensorineural. And what I mean by conductive and sensorineural is conductive is anything in the mechanical structure of the ear, whereas sensorineural means that the neurons itself can't send the right signals to the brain. And so 3 years ago, we my collaborators, Pedro Sanchez and B. Smith, the audiologists who are here doing the hearing test, came to the Alagille Alliance meeting 3 years ago. And our goal was to really test as many Alagille syndrome children as possible, just to see how prevalent is this conductive hearing loss or other forms of hearing loss. And this was because there's almost nothing in the literature about how common this might be. And we actually found that there's a much higher incidence of this hearing loss than we expected. And so of the patients we tested, I think 16 out of 23 had at least some form of hearing loss. So 2 patients had a pretty severe conductive hearing loss. 4 had a quite mild conductive hearing loss, but something that was picked up by the audiologist. And then 5 out of 23 had mixed hearing loss, means that probably there's something defective both in the mechanical transmission of sound and in the neurons, whereas 5 patients had only sensorineural hearing loss. So again, this is suggesting it's complex, that Jagged1's affecting not just one part of the hearing system, but it can be in the neurons, or it could be in these middle ear bones, or some other structure. And from one of the children who had pretty severe conductive hearing loss. So again, this is just showing, this essentially is plotting how much sound, again, at how high a sound needs to be played for the patient to have a response. And in this case, the louder sound is going down here. So having to have 50 or 60 decibels is a pretty loud sound to need. If that's the minimum loudness of a sound you need to get a response, that's a pretty bad hearing loss. And we were able to obtain one CT scan. So essentially do CT imaging of the temporal bone, and then you can see the structure of the middle ear bones. And we found that in this one patient that had hearing loss, they had a very unusually shaped incus, which is the middle bone in that chain. And actually, the malleus and incus were also fused together. And the radiographer we work with, who really specializes in looking at temporal bones in these CT scans, really said that this sort of defect was something he had never seen before. It was a really unusual, unique form of middle ear bone defect. So really, our goal is one, just get a general sort of estimate of how prevalent this conductive hearing loss is. And then in those patients that have conductive hearing loss, try to get some more CT scans to say, is it more the incus that's defective or more the stapes that's defective? And so really the summary here is that the loss of either jagged 1 or notch 2 results in defects in the middle ear bones and hearing loss. And then if you have Alagille syndrome, a hearing test might reveal either conductive or sensory hearing loss, or some mixture of the two. And that the CT scan may or may not reveal defects in the incus or stapes bones. And I think the somewhat encouraging thing is that if it is a structural defect, the structural defects tend to be easier to correct than the neural defects. And so there are surgeries available. I'm not a medical doctor. I wouldn't be able to advise someone to get this surgery, and I don't know how expensive it would be or whether insurance would cover it at this point. But there is surgery available that can use prostheses. So it's pretty amazing, but these little titanium prostheses that they can take out the abnormal bone and put these prosthetic bones in, and that corrects the hearing in not everyone, but a large proportion of patients. And this is actually quite commonly done because there's a condition called otosclerosis, which is what happens is that it's a very common form of hearing loss as people age. And what happens is you get these bony overgrowths in your middle ear, and those bony growths kind of fix the bones together so they can't vibrate anymore. And so in some of these patients, they'll remove the stapes and then put in these prosthetic stapes to restore hearing. And so there is a possibility that if there's hearing loss due to just these middle ear bone defects, that there could be some sort of surgical treatment already available for that. And then just to thank the people. These are my lab here, and I work in a stem cell and regenerative medicine institute. And I should say that a large part of my lab does bone repair and bone regeneration. These are graduate student and postdoc in my lab who did the zebrafish work. And we collaborated with Rob Maxon lab, who's another faculty member at USC who works in mouse, and then Pedro Sanchez and B. Smith who are here. And then my work in general is funded by the NIH, but this goal to really translate our findings to Alagille syndrome patients and hearing loss is funded by a March of Dimes grant that we got recently. So, and we have money to actually fly people out and pay for their expenses to come to LA to get CT scans if there's not one in their region. So, I guess if there's questions from anyone.
Does it take a special CT scan or a special radiologist to read it?
It takes a special radiologist to read it.
Okay.
And also the resolution needs to be good enough. These bones are so tiny that you really need a very good resolution scan. And so there's certain parameters that our radiologist recommends that not every center would adhere to.
You can't just go to like the walk-in MRI place and they might not.
Probably not, yeah. Otherwise, it gets kind of difficult to
Yeah.
really see these bones. So.
So our daughter has had so many different, she's flunked the drug test here and then passes the next week and the next, it's
Uh-huh.
like up and down, they'll put tubes in and out, and she just passed the one up there, it seemed fine, and
Okay.
her eardrums were moving. Before they said, no, they're retracted, they're not moving.
Yeah.
We've been dealing with this since she was probably 3 or 4 months old.
Okay.
She's 13. And it's like, at one point they were ready to put hearing aids in, and we were like, how do you know if it's
Yeah.
sensory or conductive, and, are we going
Yeah.
to the right place? And,
I mean, I would, if it's there one day and not there the other day, I would imagine it's probably not a structural defect of these bones,
okay.
is what I would imagine. I mean, you could always, has she had a scan of the temporal bone? So that could reveal something there, because the CT scan will not only reveal the middle ear bone defects, but for example, this one patient that we actually didn't do this scan when we tested her at the previous Alagille meeting. She had very severe hearing loss. She had already had a CT scan. They said it was normal, and then we took it to our radiologist, and he found this incus defect. And in addition, there was much smaller semicircular canals, so the vestibular system was also abnormal. So there could be other defects there, maybe like the eustachian tube itself is not properly structured, and so fluid can very easily get clogged up in there. So.
Well, that's what they told us from the beginning, that they're not, I guess when they're born, they're like this way, and then when you get older, the canals.
Yeah.
And hers didn't, is what they were guessing why she kept getting ear infections and fluid, and she's always got fluid in her ear. She says, it feels like I'm talking with my, when your ears get hot.
Yeah.
I mean, what are your suggestions? You like, go with the flow, or, there's some points where she struggles in school, and some where she hasn't, and, we thought, well, maybe she's just a brat and not listening.
Uh-huh.
How do you tell?
Yeah.
Is she really not listening, or not paying attention, or she really can't hear?
Uh-huh.
Is it worth investigating further, or you just, she's 13 and she's managed to deal with this long?
Yeah. I mean, I think having a scan probably, it would maybe rule out certain things that you wouldn't have to worry about. Yeah. If you say the structural stuff's normal, then you can focus more on the fluid or treating that. Yeah.
We live in Lexington, Kentucky, so I had
Oh, okay.
emailed Pedro, so there was nothing around our area research-wise on this.
Yeah. Is she doing a test here as well?
She did.
Okay.
And she said it was fine.
Okay.
So
Yeah.
that's good.
Yeah.
She just went up again. Just selective hearing at this point.
Yeah.
Thank you.
But yeah, I imagine there could be something structural with the eustachian tube that could make it more prevalent, easy to clog up.
She's fine.
Yeah.
But they keep coming out. They won't stay in.
Mm-hmm.
So. Sorry, I monopolized all the questions.
Oh, no, my dad's in the exact same situation.
How old is yours? Oh.
And does it problem kind of go away and then come back again? Is it
She'll
not?
pass them and she'll pass she'll flunk the school one every year.
Oh yeah, that's
And then
school
we
one.
take her to the day the school sends her to ESD.
ENT. Yeah.
Well, ours,
Oh,
no, ours just is the school district's level of
Oh.
retest. And then she'll barely pass that, and then they'll just come back with, okay, we'll make sure she sits in front of the class, make sure the teacher's always looking at her.
Yeah, yeah.
Make sure she doesn't sit at the noisy table.
Mm-hmm.
And so we just brought it up to our team at our big hospital, and they said, let's look more. Because I'm like, does she need a hearing aid? I mean, she wears glasses. I'm not about,
Yeah.
it's just
You don't
glasses
have
for
to.
the ear.
She's
Let's get
Sensitive
it.
to certain noises, like really sensitive to loud noises.
Yeah, fireworks and that kind of stuff.
Mhm.
Just Carly will go crazy. Or loud noises, like you'll say, do this, and they'll, what was that? What you
I was
were
doing something
doing
over
this?
there,
And she's like,
she was
stop.
but then she can't hear something. It's weird.
Uh-huh.
I know, not necessarily loud noises, but she,
Huh.
if there's any noise in the room,
Yeah.
she can't hear you.
Yeah.
Yeah. All right, well,
Did
thanks.
they fly her from